Hello, and I'm gonna go over vision right now, but I want to give you a heads up and tell you that I am gonna simplify this very much. So, if you're very interested in the physiology in terms of seeing, I'm not giving you a lot of in-depth information. I'm just gonna give you some of the basics that'll get you through the AP exam. So, starting with light, so this portion of our electromagnetic spectrum that gives rise to our sense of vision. Light moves in transverse or sine waves, and they're going to move similar to the movement of waves on the ocean. Now, I'll go over each part of this just briefly, but you'll notice for the electromagnetic spectrum, you have all these different types of waves. Visible light makes up less than 1% of the electromagnetic spectrum, and it is this range that humans are able to detect with their naked eye. I'm going to have my husband, Miguel, the physics teacher, go over some of this. So here you go, Miguel.
All right, guys. So the wavelength of a wave is the length of a cycle. So it doesn't really matter where you start, but if you measure off the length of one cycle, you've essentially measured the wavelength. So it's easiest defined from crest to crest. It can go from trough to trough, or it can go from any in-between point to a similar in-between point one wavelength away. The wavelength and the speed of light in a vacuum or air determine the frequency of light, and it's the frequency that we see as color.
This is the stuff that gets me to sleep every night.
Okay, so in the waveform above, you'll see that there are a lot of cycles there, so you know that that's going to be a high frequency wave. And down low over here in this area here, it looks like there's only 2 cycles. So this is a low frequency type waveform. The wavelength down here is pretty long. The wavelength up here, I'm going to go from a crest to a crest, is pretty short. So short wavelength, high frequency, long wavelength, low frequency. And if you multiply the wavelength and the frequency together for both cases, you get the same number. It's called the speed of light.
Some more very exciting stuff. Can you just tell them what colors they would perceive based on the different wavelengths?
I think I'll try. So if this is higher frequency, as humans, we would detect colors that are shifted more toward the blue end of the range. Blue light has higher frequency, shorter wavelength. Down here, you've got a pretty long wavelength, so you're looking at colors shifted more toward the red end of the spectrum. If you go any lower frequency than this, then you get into infrared. Infra means below. So you go below red. If you go
we don't do infrared
any
here.
higher frequency, then you should, because there are animals that could see infrared.
Yeah,
and
but
there are
we're
animals
not
that could
that
see
animal.
ultraviolet. We're not, but this is a lesson on vision, not vision of humans.
I should have been clear. Vision of humans.
So if you go above the frequency of blue light, then you wind up in ultraviolet. And bees can see in ultraviolet.
Okay, you know what? Actually, let me just make this clear this way. Don't go anywhere, Miguel. When you're looking at these waves right here, you notice they're farther apart, they're further away.
Press.
Like you have to get your notes done, read away.
That's very cute.
All right, shut up.
All right, so we kind of talked a little bit about frequency, but if you wanted to define it, it would be a measure of how frequently cycles of a wave occur. So generally it's expressed as a certain number of cycles per second, and cycles per second has a unit of hertz. It's named after Heinrich Hertz.
And that is what's determining your hue. So as Miguel already mentioned, as humans, we detect the frequency of waves as color, right? So
of visible light.
of visible light.
And if we're talking about the various senses, then the frequency of sound we detect as pitch.
But we'll do that in a different lesson.
Right. So in math class, you may have studied sine waves and cosine waves, and you learned that the amplitude of the wave tells you how high the wave is from a rest position. And it's similar for transverse waves or for electromagnetic waves. So the amplitude of the wave is the distance from rest to crest. You could go from rest to trough, but that doesn't rhyme, so... But the amplitude of the wave we detect as the brightness of light. Amplitude of sound would be volume. Amplitude of light would be the brightness. Okay, so here we have, it looks like to me, I'm trying to see if the cycles repeat, and I don't know. This might be just one full cycle of a wave, but it looks fairly complex. You're dealing with two distinct amplitudes, so you have this here as an amplitude and this guy over here as an amplitude. So this guy is roughly double the shorter amplitude waveform, but the thing that is consistent throughout is the wavelength looks fairly consistent as you move throughout. And also the frequency looks fairly consistent as well. So these waveforms occur at a very consistent basis. So a complex waveform, but you can see how some of the features like wavelength, frequency, amplitude, how we can tease these features out of this waveform.
So essentially, when you're looking at whatever color this would appear to be based on its frequency, when you're looking at these here, they would be less bright or more muted than these here. This would be much brighter in our experience.
Okay, so this looks like bio. I'm out.
Okay, so as promised, this is going to be very simplified. I'm going to tell you basically what you need to know to get through the exam, but the eye is a lot more complex than I'm going to make it sound right now. So first, you have to know that the eye captures light and focuses it on the receptors at the back of the eyeball. So the light's going to come in here, and it is going to be processed here. What's going to happen is that you have receptors in the retina and the fovea that are going to transduce the light waves into neural impulses and send them via the optic nerve to the brain to be interpreted. For the purposes of the AP exam, I'm going to tell you the parts of the eye that you do need to know that do show up on the exam. So when light enters, yep, you're going to go through the conjunctiva, but the first thing you really actually need to know here is the cornea. So the cornea is this transparent front part of the eye that covers your iris, your pupil, and the anterior chamber. And what you really want to know here is that the cornea is this convex or has this convex or outward curvature that's going to help focus incoming light rays. The light waves are going to pass through the cornea and go to the pupil. So your pupil is this adjustable opening controlled by the iris. It's a clear area of your eye, and the light is going to be passed from the pupil to the lens. The size of the pupil is also going to be controlled by the muscles within the iris. So the muscles can constrict the pupil, making the pupil appear smaller. Other muscles in the iris will dilate the pupil and make the pupil seem larger. So in low light conditions, the pupil is going to dilate. In bright light conditions, the pupil will constrict. Your iris is the colored part of your eye. So if you have brown eyes or blue eyes or whatever, the iris is what's going to give your eye that color, and it's the muscles in the iris that are going to dilate or constrict the size of your pupil. So it's going to effectively help to allow or disallow light coming in. One other thing you want to know about your iris is that while it does help to control how much light is getting through your eyes based on the amount of light that's present, your iris will also respond to emotional situations. So if I'm a really good salesman and I'm showing you some items that I want to sell, I can actually notice what happens to your pupil emotionally in response to the things that I'm showing you, and I know where I need to negotiate with you and where I don't. The next thing you need to know is your lens, and you want to know about your lens with your ciliary muscles together. Your lens is this transparent, elastic structure. It's located behind the iris, and it's going to add to the focusing that began with the cornea. But unlike the cornea, the lens is adjustable, and it's going to be the ciliary muscles that are going to help control the lens. So what you want to know here is that the lens is responsible for accommodation. Ciliary muscles are going to help with that. And accommodation essentially is what's going to help to focus the light on the back of your eye. So if you're looking at something close up, your lens will thicken and curve more. If you're looking at something far away, your lens will thin and flatten. And it is the ciliary muscles that is going to control that. And accommodation is a term that you're going to want to know. Next up is your retina. So this light that's being focused throughout your eye, ultimately the idea is to focus it along your retina so it can be transduced into neural impulses. So essentially, your light waves end up back here at your retina, at the back of your eyeball. It contains blood vessels and a network of neurons that are going to transmit neural information ultimately to the occipital lobes of your brain. So your retina contains photoreceptors. So these photoreceptors are rods and cones that are going to respond to the light input coming in. So rods are going to respond to shades of gray, your peripheral vision. They are most sensitive to dim light. And they don't see color. Rods do not see color. They don't see fine detail. You have about 120 million rods in your eye. Your cones are the other type of photoreceptor, and they are concentrated near your fovea. The cones are responsible for color vision, for fine detail. They're most sensitive to brightly lit conditions. So their function diminishes as the light dims. And you have about 6 million. So you have 120 million rods compared to about 6 million cones. So again, that fovea is located about the center of your retina, and it's this tiny pit filled with cones, and it's responsible for our sharpest vision. So these rods and cones, these photoreceptors, are going to transmit information to bipolar cells, and bipolar cells are going to either directly or indirectly transmit information to the ganglion cells. Your ganglion cells are going to kind of take all of that information that it has collected and send that information to various parts of your brain. And that information sent by the ganglion cells is going to leave the eye through the optic nerve. And remember from the physiology unit at the beginning of the year, a nerve is a bundle of axons. Responsible for carrying messages. So what's interesting though is the fact that this optic nerve has to leave your eye. It means that it causes a blind spot over here. So an area where there are no visual receptors and no vision. So there's a portion of your visual field that you literally can't see. And perception is going to kick in. So your eye is going to basically take information from the nearby areas on your retina, near to the blind spot. It's going to collect information from your other eye as well and piece it together to kind of make sense out of what should have been there. And I know that you've known this too, that when you're looking at something, you probably learned this a long time ago, but when you're looking at something, that image comes in, your eye is going to flip that. So it will come in upside down at the back of your eye, but as it will be sent not as a light wave, but a neural impulse, your brain will manage that. If we were in the classroom right now, I would let you try out some vision goggles that turn everything upside down. So you can see what that's like. But what I won't do, or wouldn't do, I guess, is let you leave them on long enough for your eyes to adjust. So if you're looking through vision goggles where everything in your world turns upside down, your brain does know that that's not actually the way your world is. And so here's this sensation perception problem. So while you're wearing these goggles, you wear them long enough, eventually everything will right itself. The problem then becomes when you take off the goggles, everything is upside down again until your brain readjusts, but it will readjust. So this perceptual process will override your sensations in a major way. And the general path, which I'm hoping you remember from when we talked about split brain patients, People who have their corpus callosum severed, remember that corpus callosum is you're connecting the two hemispheres, your lobes essentially. So you're not like severing the whole brain. So the information, if I'm looking at something, that information from my left visual field still goes to the right part of both of my eyes. The stuff from the left visual field will ultimately end up being perceived in the visual cortex of my right occipital lobe. The stuff from my right visual field of both eyes will go to the left side of both eyes and will be interpreted in the visual cortex and the left occipital lobe. Now, if you look at the path it takes, I just want to remind you of a couple of things. So obviously, you know, any information coming from the left is going to be ultimately end up being perceived by the right hemisphere. Anything coming in from the right is going to ultimately be perceived in the left hemisphere. But each hemisphere will share that information with the other side, unless, again, you have a split brain, in which case you can't transfer the information that way. But if you remember and think about this, me telling you the part of the brain that is like your sensory relay station for all of your senses except olfaction. Hopefully you already remember what that is. That's your thalamus, right? If you follow this path here, the optic nerve, where you have this information leaving your eye, to the optic chiasm, where ultimately at that point it's going to switch. Like everything that needs to go to the left will go to the left, everything needing to go to the right will go to the right. That's at the optic chiasm. It'll follow the optic tract to the lateral genucleate nucleus, or the LGN. That is part of the thalamus. So I don't want that to seem more confusing. This right here, part of the thalamus. This right here, part of your occipital lobes. And this is essentially what the path would look like from your light seeing light waves up through your brain perceiving those light waves. Explain this, make sure you know it. That accommodation is how your lens is going to focus light to the back of your eye. The idea is that you should have your sharpest vision right at your fovea, right landing squarely in your fovea, right at the center of your retina. And when the lens is focusing, if it's doing its job correctly, again, stuff close to your face, it's going to cause your lens to curve and thicken. Stuff further away, it's going to thin and flatten. It's going to stretch a little bit. And that's going to help you with acuity. So acuity is how sharp is your vision. That's your acuity. 20/20 vision would mean that at 20 feet away, you can see the exact type of detail that somebody who has normal vision can see at 20 feet away. If you have 20/50 vision, it would mean that at 20 feet away, the detail you see is the same as what somebody with normal vision would see at 50 feet away. The mark for blindness would be 2200 with corrective lenses. And nearsightedness or myopia would be a visual acuity problem. So you're having trouble with the sharpness of your vision. And it can result from a few things. Your cornea and your lens focusing an image in front of the retina instead of on the retina. You can have a deeper than normal eyeball in relation to your lens. So your cornea, it could be too sharply curved. There's a number of things that could cause this. But essentially, when that light, the lens, your cornea and your lens are supposed to help focus that light on your retina, and if that happens before your retina, you end up nearsighted. You see there in this image that's focusing the light before the retina. That ideal visual acuity is not happening on the retina, it's happening in front of the retina. And farsightedness, that ideal spot is going to be behind the retina, not on the retina. And this is what that would look like. So as the light waves come in between your cornea, the shape of your eye, the lens, when that light gets focused, that ideal spot for visual acuity happens beyond the point of your retina. Dark adaptation, when you walk from a bright area outside, let's say, to a dark area inside, you're momentarily blinded. The bright light is going to essentially bleach the rods, making them temporarily non-functional, and rods are what you need in order to see in dimly lit situations. So you need this rapid shift from cones to rods, and your rods aren't prepared for that. So picture going into a movie theater and it's late. You're meeting your friends, you're late. Like you walk in, the movie theater is just pitch dark and you can't see a thing. Then you're looking all around trying not to trip over people as you find your friends. You sit down, you're fine. And then, like 20 to 30 minutes into the movie, you look around and you can see everything just fine. Well, yeah, it takes a second or two before the rods are functioning enough to see, but it does take about 20 to 30 minutes before you've totally adjusted from the light to the dark situation. Light adaptation takes about 7 to 10 minutes. It's the work of the cones. As we age, the process is going to take longer. So picture this. You have room darkening, blinds in your room, and you're sound asleep, lights off, and all of a sudden you freak out because you're like, oh my gosh, did I oversleep? But it's totally dark in your room. So you check your cell phone, and as you do, the light on the cell phone feels like it blinds your eyes. Light adaptation. Give it a little bit of time and it won't be bothering you anymore. Notice how your visual acuity improves as time passes. So the trichromatic theory is one of the theories that helps to explain how we see color. Trichromatic theory is also called the Young-Helmholtz theory because of two people that worked on this theory. Actually, Helmholtz was building off of Young's work. And I know if you go back to like elementary school, kindergarten, probably, you learn about primary colors of pigment. Primary colors of light are not the same. So the three colors that you would use to make up all other colors that we perceive in your eye, taken care of by your cones, would be red, blue, and green. And you just want to make sure that you remember those three colors, red, blue, and green. Those three colors will make up all the other types of combinations that you see. It doesn't explain everything about the perception of color, but it does help to explain what's happening in the retina. And that's key. Remember, this is happening with the cones, which means it's a photoreceptor in your retina. So Ewald Hering came up with this theory, the opponent process theory, that explains how we perceive color in the brain. So this processing happens in your brain, in the lateral geniculate nucleus, or the LGN of your thalamus. And it's essentially saying that when one color is stimulated, the other one is inhibited. So there are pairs of colors that are going to help with the perception of color. And you have to remember these pairs. Obviously, it's easy to remember the title, opponent process. You've got pairs of colors. It's easy to remember if you think of it like this. Red and green, Christmas colors, whether or not you celebrate Christmas. They're still Christmas colors. Yellow and blue happen to be your school colors. And black and white, like the best cookies ever. Those black and white cookies, those are amazing. So if you are a trichromat, it means that you can see all the colors on the visual spectrum. If you're a monochromat, it means that you cannot see anything but black and white. And dichromats are people that can see some colors but not others. So you notice that people who are colorblind often have trouble seeing like one type of color. So they might not be fully able to see red or green or blue light. So if you have red-green color blindness, you'll probably confuse blue and purple because they can't see the red elements of the color purple. The opponent process theory, again, in the brain, in the lateral geniculate nucleus of the thalamus, it helps to explain after images. So if you were to take this image, and you can if you like, just pause on the video for like 20 seconds and just stare at that center cross, you'll notice, remember the pairings that I told you, red and green, black and white, yellow and blue. So if you stare at this long enough, you're going to tire out the neurons that have been firing for these colors, and you will see its opponent color. So stare at this for about 20 seconds. If you pause on here, then look down at a blank sheet of paper, and what is green will turn red, what was black will turn white, and what is yellow will turn blue, and you will see the American flag.